Welcome to this lecture on my research on the FTO gene and its associations with depression and diet, specifically in females. Okay, let's start with the FTO gene. This is the fat mass and obesity gene and was one of the first genes to be linked to obesity via GWAS or genome-wide association studies in 2007. The A allele of the SNP RS9939609 is associated with higher BMI and obesity risk. The common allele for RS9939609 is T. The FTO gene is highly expressed in the hypothalamus and plays a role in energy balance, appetite, and eating behavior. Now, we know it is common to see obesity and depression together. They are considered comorbid, which means they often occur together. And research has indicated that there is a bidirectional relationship between the two. That means if you are obese, you are more likely to be depressed. And if you are depressed, you are more likely to be obese. Teasing out the causal factors behind this relationship is complicated. Since we already know that FTO is associated with obesity, I decided to explore whether the FTO gene is also associated with increased risk for depression. All right, research also shows that FTO variants influence eating patterns and that diet quality itself is associated with depression risk. I plan to examine the FTO, depression, and diet connection more closely. When doing research, it is really important to figure out where gaps exist in previous research. And one of those gaps involves looking at sex-specific associations. Are these associations between the FTO gene, depression, and diet unique depending on biological sex? In my analysis, I started with 26 FTO SNPs. Remember, SNPs are specific locations within the gene with a single base alteration. These SNPs would have begun as a mutation, but once they are seen in at least 1% of the population, it is no longer called a mutation, but is now called a single nucleotide polymorphism, or SNP. Of the 26 FTO SNPs that we started with, only 22 of them met actual quality control standards, and so we eliminated 4 of them. All of them are located in the first intron of the gene, so they are in close proximity to each other. And then also the FTO gene is found on chromosome 16. Okay, let's look at how I used genotype in my analysis. When I did my chi-square analyses, I use this setup right here. Logistic regressions work a little bit different, and I did do some of those. And we're not going to cover logistic regressions, but for my chi-square and odds ratios, I use this setup. With each SNP, there are 3 possible genotypes. Using RS9939609 as an example, the alleles associated with this SNP are A and T. So a person could have the genotype AA, AT, or TT because there's 2 copies of each chromosome, which means 2 alleles for each SNP. The A allele is considered the risk allele that is associated with obesity. I decided to set my chi-square up where the first row is people who are homozygous for the risk allele, or in this case, that would be AA. And then the second row is everyone else. So that would be people who have the AT and TT genotype. So I had all those people, added them up, and that was row 2. The columns would be either depression, which I used the total score from the PHQ-9 questionnaire, and I used scores of 10 or higher. I mentioned that in the last lecture, that score of 10 or higher kind of is a good threshold for determining depression. And then the other column was people without depression, and they had scores on the PHQ-9 from 0 to 4. When doing the diet analysis, I will use a yes and no for the diet questions in the columns instead of the depression status. So let's take a look at what that looks like. So here you see the count or contingency tables with the genotype groups. No, this is a different SNP. It's RS 1121980, so not the same SNP from the previous slide. The alleles for this SNP are A and G. And then you can see that the depression groups are labeled 10 plus and 0 through 4. Hopefully you can see where I'm looking. This is below the bar graphs. There's one for females, which are on the left, and there's one for males that are on the right. The basic question being asked in this analysis would be, is RS 1121-980 associated with having depression in males and or females? More specifically, are people who are homozygous AA associated with having depression in males and or females? I ran this analysis in a coding platform called R. It's kind of like Python. And that just allowed me to loop it through all 22 SNPs in one run rather than having to do them each individually. The example here shows RS 1121-980 with females on the left, males on the right. The blue and orange bars are just a visual way to see how the data counts look. I converted the counts into like a percentage. So just by looking, you can see that the males have very similar numbers of people with and without depression in the different genotype groups. So one of the bars, is the AA group. One of the bars is AG plus GG. And you can see that the yes and the no, or the orange and the blue sections, are very similar in the males. Now, if you look at the female bar graphs, which are on the left, they have a more noticeable difference. So just visually looking doesn't tell us anything for sure, but it can give an idea of if there's anything going on there. So let's take a look at the actual p-values from the chi-square. You can see those below the contingency table or the count tables, and I underlined them in kind of a deep red. The males show a p-value of 0.88, which is not significant. This suggests that it doesn't matter what their genotype is at this SNP, their risk for depression is basically the same. You may notice there is no odds ratio because I only performed an odds ratio if the p-value is significant. Now look at the females. The p-value is 0.006, which is quite significant. That means that there is an association between genotype and depression in females at this SNP, specifically with people who have the AA genotype compared to the AG and GG group. The odds ratio of 2.53 suggests that females who have the AA genotype at this SNP are 2.53 times more likely to have depression than females with the AG or GG genotype. I want to remind you that the interpretation looks at the first row, which is AA, and the first column, which is 10 plus, and that's the PHQ-9 score of 10 or higher and represents having depression. So I will interpret the odds ratio in the context of having the AA genotype and having depression compared to those with the AG and GG group. So again, females with the AA genotype are 2.53 times more likely to have depression compared to those with the AG and GG genotypes. I could also say females with the AA genotype are 2.53 times more likely to have depression than those with at least one G allele. You can kind of look at that. The AA genotype has no G alleles, so I could lump them that way. I also want to point out that we have an odds ratio greater than 1. We talked about in the last lecture how that matters. Let's say, for example, the odds ratio was 0.5, so less than 1. The terminology would change to like females with the AA genotype are, less likely to have the depression. So like 50% less likely. That 0.5 would represent a 50%, or 0.5 times less likely. So just make sure you understand more likely is correlated with an odds ratio greater than 1. Less likely is going to be correlated with an odds ratio less than 1. Okay, now let's look at the results of the chi-squares that I did for all of the FTO SNPs. So now we're not looking at count tables. We're not actually looking at the p-values. We're looking at a bar chart that represents the p-values, and this is just significant p-values. So it doesn't actually have all 22 SNPs that I've done. It just is showing us the ones that are significant. But what we can see is the difference between the p-values for total cohort, those are in blue, and the p-values for just women only. The horizontal dashed line represents the significance threshold. And so if the bar goes above that line, then you have a significant association. And that would be based on the p-value that came from the chi-square. You can see that there are more associations between genotypes and depression in females, the orange bars, than in the total cohort, which is the blue bars. And I want to point out that RS9939609, I have a green line drawn to that, which we've mentioned before, because it has that strong association to obesity. It also has the lowest p-value in association with depression. Sorry. After identifying the associations exist, we had to perform the odds ratio. Again, remember the associations tell us there's something there, but it doesn't tell us what's happening. It doesn't tell us like the strength of the association or what it's actually doing. So we could see the direction and effect of the association with the odds ratio. This is what we call a forest plot, and it shows all the odds ratios for our significantly associated SNPs, and this is in females. So I'm not showing the ones that were total cohort. Now we're just looking at odds ratios in the female cohort. You can see that all of the associations have odds ratios greater than 1, which means that all of these alleles are associated with an increased risk or odds for depression. Now, this one is representing a logistic regression, not the chi-square. So it's similar, but not quite exactly the same. But the thing that the logistic regression could do is tell us how much of an increased odds is going on with each of the risk alleles. Instead of like with the chi-square, I set it up where we were talking about people who are just, homozygous for the risk allele. This one tells us like a per allele adjustment. And so what I'm showing you is the increased risk per allele. So look at RS9939609 again. It has the highest odds ratio. Again, I drew a green line there. The risk allele for this SNP is A. So the way we would interpret this is that the odds ratio is telling us that for each A allele a female has, the risk for depression increases by nearly 2 times that of those without the A allele. So if you have 2 A's, then your odds are even higher. All right, I wanted to look at this haplotype really quickly. I'm not going to dig too deep into it, but you guys did get introduced to this concept earlier in the semester. So a haplotype is just a set of nearby variants that are inherited as a package, okay? Now, I showed you that all of these variants were in the first intron. They're really close to each other. And so the likelihood that they would be inherited together is high. They're in a haplotype. So we wanted to look and see what was going on there. And we found 2 common haplotypes. We'll call them packages. 2 common packages across 5 SNPs. And when we tested them, one package increased the odds of depression. That's to the right of the dashed line. And the other decreased it, which is to the left. This tells us that groups of variants can act together. And this is one reason why genetics can get tricky, because we saw that with individual SNPs we had increased odds. Here we see that certain ones in combination are increased odds. There's a lot of variations that can go on, but I wanted you to see the idea of a haplotype in action. So we have two haplotypes represented here that were found within our cohort. One of them is associated with increased odds of depression. One of them was associated with decreased odds of depression. Again, odds ratio on the left, that one is lower than 1, so it's decreased odds. Okay, now let's take a look at the nutrigenomics part. That's our topic this week. We have already shown that the FTO SNPs are associated with an increased risk for depression in females. So here we have a heat map that's showing how genotype and diet can interact with depression. The darker the blue, the stronger the association there is. In other words, if the square is white, there is no interaction between genotype, depression, and diet. The black asterisks mark the interactions that are significant. Males are on the right and have no significant interactions, but females, which are on the left, actually have 2. The interaction involves 2 different SNPs and 2 different diet traits. And again, this just shows that there is a significant interaction, but it doesn't show what the interaction is. So let's add the odds ratio and see what we get. Here we have a couple of bar charts. They're representing those 2 squares on the previous slide that had the asterisks in them. So our 2 significant results, now we've applied the odds ratio calculation. The 2 SNPs, the one on the left shows that for each risk allele, which is A, at RS 6499640, the risk of depression decreases if that person eats vegetables daily. And then the chart on the right shows that for the risk allele carriers at RS 9939609, which is also A, eating pasta or rice more than 5 times a week increases the risk of having depression. So the way what that means is if, let's say you had an AA at that RS 9939609, that would mean that if you eat a lot of pasta and rice, you are increasing your risk or odds of having depression by quite a bit. The odds ratio is 3.39, which is pretty high if you had 2 risk alleles, which was AA. On the flip side, with the other one on the left with the vegetables, say you had 2 risk alleles there, which also happens to be AA. If you were AA and you were eating vegetables daily, you're actually reducing your risk of depression, and it's all the way down there at 0.19, which is quite significant. That's a really, really nice reduction in your odds. And so this is a perfect example of how understanding what genetic makeup you have can help determine your risk for a certain phenotype, in this case, depression, and then how diet could be used to reduce or increase those risks. After seeing this small snapshot of the research I'm doing in genomics, I hope you can appreciate all the work and effort that is required for understanding our genome better and then identifying ways to work with our genes to improve our health. Our genes provide a blueprint for our health, but we now understand how intertwined our health is with our genetics and also our environment, from where you live, what your ancestry is, your parents' health, pollution exposure, lifestyle choices such as diet, exercise, smoking. The list goes on and on. Every scientist spends hours working on one tiny piece of the puzzle in the hopes that with all the effort combined, we can finally get a big picture and gain enough knowledge that we can find realistic ways to prevent disease and improve health. There are so many different components that have to be considered. We have really just scratched the surface in this class, but I hope that you've enjoyed learning all about the human genome and all the research that has gone into understanding it and that continues to explore and identify new things and technologies meant to improve our lives. Thank you.